Hello everyone, welcome to Practically's NEET Bio class, and today we're going to discuss about enzymes. Okay, so this is a very important topic because there are lots of things actually involved in it. Till now we were discussing about plants, animals, so many things. Okay, but this particular topic is actually a lot of chemistry is also involved in here. Okay, so this is bio. Okay, don't doubt, this is bio, this is pure bio, but there's a lot of chemistry involved in it because this enzyme does a lot of chemical works. Okay, so the whole catalysis and the changing the reaction rates and everything, so many things we learn in chemistry. This is one thing which actually affects real-time reactions in our body, in certain chemical reactions, etc. A lot of things are actually influenced by enzymes. Okay, so this is the last class for this particular topic, that is biomolecules, and I have saved the best for the last because this is quite interesting. If you read it properly, if you actually pay a little bit of attention into it, you can find a lot of things, a lot of valuable information which might be helpful for your exams as well. Okay, so let's get started. Before that, I'll talk a little bit about us. We are from Practically, and we have an app, a very interesting app which runs for grade 6 to 12. Okay, you can find a lot of stuff in there, a lot of fun stuff which you can actually use in your life, for your studies. Okay, it'll look like a game, but it'll be helpful for your studies as well. You can also follow us on our Facebook and on Instagram, and do not forget to like, share and subscribe to our YouTube channels. Okay, we have a new YouTube channel as well. You can check that out. Okay, that is specially dedicated to 11th and 12th grades, and you can subscribe to that channel as well, and that will be concentrated towards those particular grades, 11th and 12th grade. So do not forget to subscribe to that as well. Okay, so lot of talks. Now we'll directly jump into the class. Let me see if somebody has joined. Hi Sumit, welcome to the class. Okay, I hope you enjoy this class. Okay, so now, so what are enzymes basically? Just think it like that. Okay, so you are cooking something. Suppose okay, I'm a non-vegetarian. Okay, my mother cooks mutton. Okay, I love mutton, and my mother cooks mutton. Now the thing about mutton is it takes a lot of time to actually get cooked. Okay, so what my mother does, my mom, sometimes she puts pieces of papaya in it. Okay, now papaya has a lot of enzymes in it, if you don't know. Okay, so when papaya, you put a little bit of papaya into that cooking thing. Okay, when my mother is cooking, my mother puts a lot of papaya, not a lot, a little bit. Okay, so she puts it. What it does is it actually helps in the cooking. Okay, what it does, apart from giving a little bit of texture and a little bit more of what to say taste, what it increases the rate of the reaction. Okay, so that's what it does. So these enzymes, now papaya is full of enzymes. Okay, so when you put it into a mutton curry, what it'll increase the rate of the reaction. The mutton will cook a bit more faster, less amount of cooking time. That's what my mom wants. So that is how it is. Now similarly, in day-to-day work, in life, also in our body, we need some catalysts. Okay, what is a catalyst? A catalyst is something which pushes, okay, it literally pushes the rate of the reaction so that it becomes a bit more faster. Okay, so that is what an enzyme does. It is a biological catalyst which influences the speed of the biochemical reactions. Okay, so I hope you understood it. Now, one second. Okay, so as I said, a biological catalyst which influences the speed of the biochemical reaction. As I said, so when a mutton is being cooked, when you put papaya, okay, it behaves as a catalyst. Okay, so what? Mutton cooks faster. That is how it is. So now the basic idea of enzyme is all enzymes are proteins, all of them. Okay, so we learned about proteins. Proteins are made up of small constituents such as amino acids. Okay, one amino acid sits after the another. Okay, and they form into a kind of a 3D kind of a structure, one onto another. We have alpha helices and we have beta pleated sheets and so many things there are. And okay, so they are all proteins, and the structure of enzyme is also like proteins also. So enzymes are basically they have proper structure. They have alpha helices, they have beta pleated sheets, they have a proper 3D structure. Okay, so when you actually like you want to draw it or something, so it will look almost in a 3D drawing. If you do, it will almost look like this. Okay, lots of amino acids coming together forming enzymes. Now the unique thing about enzyme is they have their own substrates. So they will attach to this particular substrate only. Okay, no other substrate will work. Substrate will help the enzyme to get activated. Okay, the enzyme will only activated means the enzyme will only work. Okay, they'll only work when they have this substrate. Okay, so suppose now there are ribozymes. These are nucleic acids, RNA. Okay, especially not DNA, this RNA that behaves like enzymes. So what else do we know about enzymes? Now enzymes form tertiary structure. This I said because they're proteins in nature, right? So of course they'll have 3D structure. Okay, they'll have a tertiary structure because what is it? We have primary structure, that is just amino acid, one amino acid after another. Secondary structure, that's just alpha helices and beta pleated sheets. Tertiary structure, okay, just alpha helices and beta pleated sheets, they just bunch onto each other and they form a tertiary structure. We have quaternary structures also, where it's just all the structures come together into a lump. Okay, so now what the specialty of enzyme is, they have some crevices, some areas, okay, particular areas where this substrate, this unique substrate that will fit there only. Okay, so suppose this is an enzyme. Okay, so they have a particular substrate, they have a particular area. So this area is a crevice. Now the substrate will look somewhat like this. Okay, suppose this is the substrate. So they'll just fit onto each other. They'll just fit onto each other because they're unique to each other. It's like a lock and a key. I have a lock. The lock will get opened only by one particular key. Why? The structure is like that. Similarly, this is also the same thing. So this is an enzyme. This is a substrate. This will attach to each other. Nothing else. They'll only attach to each other. That is how it works. Now, through their active site, they catalyze the reactions. Only after the substrate has actually jumped onto the enzyme, they'll get activated and then they'll do their work. They'll catalyze reactions. Okay. Now, there are some enzymes. Now, most of the enzymes actually work at an optimum temperature. The optimum temperature for most of the enzymes works around, suppose, say, some 25 to 35, 37 degrees Celsius. Okay, most of the enzymes. So when it is high temperature, such as 40 degrees Celsius, the enzymes tend to get damaged. Their protein structure, they get denatured. Okay, but there's always an exception. As we know, in biology, we have a number of exceptions. So there's enzymes which actually survive 80 to 90 degrees Celsius as well. Okay, and one of the best examples is Taq polymerase. Okay, so Taq polymerase is actually produced by, where's my cursor? Yeah. So Taq polymerase is produced by a bacteria called Thermus aquaticus. Okay, So this thermus aquaticus produces an enzyme called TAC. You know where this TAC is used? TAC polymerase is used in PCR in reactions. What is PCR? PCR is polymerase chain reaction. We know about PCR. Why do we know about PCR? Because PCR tests are the ones which actually help us diagnose COVID-19. Okay, so TAC polymerase is one of the polymerases, one of the enzymes which actually is used in the reaction for PCR. Okay, so PCR is a reaction where different DNAs come and they are, we'll learn about PCR more in the next classes. Okay, but PCR is something where we get small amounts of DNAs and we make it into a bigger chain so as to diagnose all kinds of infections such as COVID-19. There we use this enzyme called TAC polymerase, which actually can survive up to a very high temperature of 90 degrees Celsius, actually 94 degrees Celsius. Okay, so this is an exception. Okay, so now first we'll talk a little bit about the chemical reactions and then we'll talk more about the enzymes. Now we know that chemical reactions are actually of two types. There's a physical change which we can see with our eyes. Okay, so you take an ice cube you take and you keep it in a hot room. Okay, no fan, no AC, no nothing. It's a hot room. And within just minutes, you can actually see that the ice has melted into water. Okay, in front of your eyes, you see ice has melted into water. Done. That is a physical change, something which you can see with your eyes. Then there is water vapor also, where you just boil water, water becomes vapor. These and all are physical reactions. Okay. Now there are chemical changes also. This actually happens inside the constituents, inside the compounds. So what happens? In this, bonds are broken and new bonds are formed. So there is a compound, okay? Suppose one element, another element form together, they make a compound. Now there is a reaction which is happening around them. Okay, this bond breaks and this element goes and attaches into another element. How? Suppose, look at this reaction. So this is BaOH2, that's barium hydroxide, which when added to H2SO4, that is sulfuric acid, it becomes BaSO4 and 2H2. So what happened? This barium hydroxide, this barium thing actually came and attached itself to this sulfate. Okay, so the bonds break here and it attaches here. So these are all internal. This is chemical. Why? This is internal. You cannot see this happening in front of your eyes. In front of your eyes, it won't break and go and attach itself to something else. No, these are internal. That is why it is chemical. So there are two types of reactions now we know. There is physical change, there is chemical change. Now, so how does a reaction work? Okay, the question should be like that. Why am I saying this? Because this and all are related to enzymes only. Please don't get irritated, ma'am, we know this and all. No. So these are just the basics so that you understand how enzymes work. Okay, so now the rate of the physical or chemical process is the amount of the product formed per unit time. Okay, so that is given by dou P by dou T. So P is the product and T is time. Now rate is called the velocity if the direction is specified. If it is something attaching to something, okay, per unit time, when there's a time involved in everything, okay, that gives the idea of velocity, the speed. Okay, so rates of physical and chemical processes are influenced by factors such as temperature, it can be pH also, it can be other things also, and the rate doubles or decreases by half for every, because the environmental, these are the environmental changes, not the environment, the environment of the constituents. Okay, so the environmental changes. So accordingly, suppose it is an enzyme. Okay, so enzyme 1 is there. Okay, and suddenly what you do, you increase the temperature by what? By some 10 degrees Celsius, and it becomes 50 degrees Celsius. It was 40, now it becomes 50 degrees Celsius. So what happens? The enzyme denatures and your reaction rate decreases, because it has denatured. It has become 50. Now it cannot work. So it has decreased. So everything, all the, be it enzymes or any other catalyst, okay, so all these catalyst enzymes, they depend on the environmental factors. Okay, so now that and all done, now let's talk about talk about enzymes. So the rate of enzyme catalyzed reactions is very, very high. So what happens is, when you add something, a bit of an enzyme into a reaction, it not only doubles or triples, it actually becomes like very, very high, astoundingly high it becomes. For example, see this reaction. So there is an enzyme called carbonic anhydrase. This is the fastest enzyme in the world. Okay, in the world, this is the fastest enzyme. So, and it increases the reaction by 10 million times. So if suppose there is carbon dioxide and water, and it forms carbonic acid, that is H2CO3. Okay, so there is no carbonic anhydrase, somehow it will form carbonic acid. But when there is carbonic anhydrase, it becomes around, suppose in the absence, it is around 200 molecules. Just 200 molecules are formed in an hour. But when it is present, around 600,000 molecules are formed in an hour. So see the temperatures. Without the enzyme, it is just 200. Okay, and with carbonic anhydrase, it is 600,000 per hour molecules. So you think how fast it is. Okay, because within an hour, it is forming 600,000 molecules. Instead of that, if you don't put an enzyme, it will form only 200 molecules. So each of this is just carbonic anhydrase. This is the fastest enzyme in the world. But all of the enzymes actually work very fast. And all our body things which are happening, all the reactions in our body, be it glycolysis, gluconeogenesis, respiration, everything actually takes part with the help of enzyme. See, glycolysis, glucose is converted into pyruvic acid. Okay, and there are around 10 different enzymes actually help in that reaction. The same for gluconeogenesis, because that is just the opposite. There is respiration in plants, there is photosynthesis, a lot, dark reaction, light reaction, a lot of them actually use a lot of different enzymes. And only because there are enzymes helping in these kind of reaction is why our bodies work so fast. Okay, when we eat, there are different kinds of enzyme actually helping our body, or different organs, to actually process the food, to help. It helps the liver, it helps the small intestine, it helps the large intestine. helps the stomach and everything. It's the work of the enzymes which actually makes us actually live so nicely, healthily. Okay, and it's not just humans, plants, different animals. Everybody has their own set of enzymes which actually work beautifully. Okay, so let's see who else is there. Hi Neha, good evening. Okay, so now how does the enzyme work? As I said, the first thing which I said is enzyme is very unique. Okay, it has a unique structure. Why is it a unique structure? Because enzyme will only attract their own substrate. And without substrate, it won't work. It's not that only enzyme, okay, working, gone, done. No. Only if they have their own substrate, it will work. Okay. And it is like a lock and key model. Okay. So literally a lock and key model. See. So this is the substrate. Our key is the substrate here. And there is a enzyme. The lock is the enzyme. Only when the lock and key complex has formed, the door opens. है ना? So similarly, this is an enzyme. It has a specific active site where the specific substrate will actually come and join. So now it has formed the enzyme substrate complex, that is ES complex, and it is tightly bound. Suppose this is the enzyme and this is the substrate, it is tightly bound, and then only the reaction starts. Okay, so enzyme, okay, let me just see. Enzyme, substrate, tightly bound each other, then only this compound will work. Okay, if it is not there, if the active site is open, it won't work. Only after the substrate has come and it has joined, then only it's working. Otherwise, it won't work. So it is like a, will the lock open without the key? It won't open, right? How much ever you try, it won't open. Okay, until and unless you come and break it, that is a different story. But only if you have the key, you turn it and it will work. Similarly, enzymes will work when there is a substrate. Okay, so now that is enzyme and substrate. What happens after the enzyme and substrate has joined each other? Now, because the enzyme and substrate have joined each other, because of that, the products will form. Okay, because there is a joining, the substrate, okay, somehow it converts into the product. Okay, see, the active site breaks chemical bonds of substrate to form the enzyme product complex. Okay, so this is the substrate. Okay, enzyme has joined it. It contours it in a way that the product is formed. Then finally, when the reaction has completed, the product will go away. Okay, so the enzyme releases the products and free enzyme is ready to bind to other molecules of the substrate. So reaction complete. Now I am released. One of the best examples here I have given is the hydrogen peroxide and peroxidase enzyme. So hydrogen peroxide here is the substrate. Okay. Peroxidase here is the enzyme. Peroxidase has been waiting for H2O2 or hydrogen peroxide. It's been waiting there. So suddenly H2O2 comes, and see the unique structure it has. It will only, this peroxidase will only bind to hydrogen peroxidase, nothing else. Okay, so it will bind here, it has bound, and it forms the products. It contours it in a way that it forms the products. Products are what? There is water and there is oxygen. Okay, and then they go out. Now it is ready for another H2O2 molecule, the next H2O2 molecule, it is ready for that. Okay, so that is how it works. See, very easy. So I have H2O2. This is my H2O2 molecule. And I have a peroxidase enzyme. This attaches here. Okay. And because of that attachment, because the enzymes have formed, so it forms hydrogen and oxygen. Okay. Sorry, water and oxygen. Sorry. It's water and oxygen. And then it is released. Water and oxygen is released. Now again, this enzyme is again ready for another molecule, another H2O2 molecule only, because unique structure. Okay, it will attach to only H2O2. So that is how actually the enzymes work. Okay, so now done. Okay, this is how enzymes work and everything. Now I'm okay. Now I have to understand why or how these enzymes are actually helping the catalysis, helping the catalysis of the reactions, and how is it pushing it. Okay, so let us consider an example. Okay, let me just check if there are any comments or anything. Okay, no. Okay, let's do it then. Suppose I am starting my day. Okay, now I woke up right now and I'm starting my day. So the moment I open my eyes, I think, okay, I have a lot of work to do. Okay, today I have a lot of work to do. I have to teach. Okay, I have to teach some students. I have to go to the shop to buy some products for my home. I have to finish my work. Okay, I have to finish my household work. Okay, I have to go meet my parents. Okay, so I have a very eventful day. Okay, I have a very eventful day today. Okay, now what? Now I don't feel like getting up. Okay, I don't feel like, although I have a very important day today, I don't feel like getting up. Why don't I want to feel like getting up? Sometimes I don't want to feel like getting up. Although I have a very eventful day today, no, I'm not getting up. So what I do, I motivate myself. Okay, I motivate myself. Okay, come on, let's just get up and just start our day and we'll do it. Okay, I'm motivating myself to, okay, come on, let's just start the day. We'll do it. So that motivation is what the enzymes do. Okay, very simple. It is just the push, that final push somebody gives so that the reaction happens is what the enzyme does. Okay, so now that is in story terms. In scientific terms, what it means. Now, as I said, I am waking up and I think, okay, I have a lot of work to do today and I need some energy. That energy I don't have. That energy for me is the activation energy. Okay, I am fully drained out because I worked the last day also and I'm fully drained out, but I need some energy. I have a lot of work to do today also. So I need some energy. That energy is the activation energy. Now, in scientific terms, in an exothermic or endothermic reaction, the substrate must go through a high energy state, that is the transition energy state. Now, just think this. Now, any reaction, exothermic, endothermic, where heat is formed or heat is absorbed, whatever, not important. In any reaction, what is required? A push is required. Okay, so that you are all excited. Okay, suppose I am waking up, I have to get excited for the day. Okay, that excitement phase is the transition energy state, where I am bound to, well, like I'm not started, but I am about to start the work. So that thing is known as the transition energy state. Therefore, activation energy is the difference between the average energy of the substrate and the transition energy of the substrate. In layman terms, when I'm waking up and I'm thinking about all the work and I'm excited to do that work, that difference, okay. So this is where, suppose this is where I'm waking up and this is where I have to be excited. So this difference is the activation energy. Okay. So what the enzymes do is, okay, so enzyme, as I said, what is enzyme? That motivation, okay, that motivation is given by the enzymes, and the reaction actually does not require a lot of activation energy, okay, because it is pushing it a bit more, it is pushing the activation energy a little bit more, so it does not require that much, it requires less amount of activation energy, okay. So if the product is at lower energy level than substrate, the reaction is an exothermic reaction, okay, and it is just the opposite for endothermic reaction. It requires no energy to form the product, okay. So leave all this, just understand this. What enzyme does is, it performs the critical task of lowering the activation energy. Just understand this. So I have woken up, okay, I need to excite myself, okay, let's go, let's do it, let's do it, let's do it. I don't have the energy. What a motivation does is, it just pushes me. I don't need to be that excited, I just need to be a little excited so that I can just do the work, okay. I don't need a lot of energy for that, I just need a little bit energy so that I can finish the work. That is what the enzyme does. Enzyme is like the motivation. So what my motivation tells me? It tells me, okay, you don't have to be very energetic, you just need that little bit amount of energy so that you can finish your work. So enzyme just pushes or catalyzes a little bit of activation energy, okay, so that the products are formed. Okay, so you don't have to go to that high level of activation energy, not required. Just need a little bit of push and you can finish your work. That is what enzymes teaches us. Okay, so I hope you understood. So for example, see, what I did is, see here, carbon dioxide and H2O. So when I put carbonic anhydrase, okay, it forms the carbonic acid, okay. It does not require a lot of activation energy, only it requires just this much activation required. Without enzyme, you need a lot of activation energy, okay. Without that motivation, you need to be like pushing yourself, okay, not doing like that, whatever, okay. But with the enzyme, you have the courage and the ability to just push yourself through the day. So enzyme is that final push which actually forms the products, okay. So I hope you understood. Now, factors affecting enzyme activity. So as I said, all the reactions are actually affected by all kinds of environmental, their environment, not ours, environmental, like environmental factors. Okay, so the best ones are, okay, sorry, my pen tab actually malfunctioned. Okay, so what happens? Like temperature and pH. As I said, enzymes will work only up till a particular pH or up till a particular temperature only, which is their optimum. Okay, both temperature and pH, it has to be optimum only. Okay, it cannot be too low. If it's too low, it won't work. It will be inactive. Okay, be it temperature, pressure, pH, it cannot be too low. It will be inactive. If it is too high again, then what will happen? It won't work only, it will get denatured, it will get destroyed. Protein component, no, it's a protein component. Very high, it will start breaking here, there. So it won't work only. Now, inorganic catalysts, actually inorganic catalysts, which are not organic in nature, not proteins, they actually work at high temperature and pressure. But enzymes, okay, they'll work only at a particular, as I said, most of the enzymes actually work from 25 degrees Celsius to 37, 38 degrees Celsius. Apart from what? Apart from Taq polymerase, which is produced by, just now we discussed about it, Thermus aquaticus. Okay, so Taq polymerase, that is Thermus aquaticus, it produces Taq enzyme, which is actually used for different kinds of reactions such as PCR and all. This one actually survives for 94 degree, 95 degrees Celsius easily, because the bacteria is actually living in a very high temperature based place. It lives in hot springs, etc. Okay, and the enzymes which are formed, which although proteinaceous in nature and everything, they can survive high temperatures easily. But this is an exception, not the rule. The rule is it will only survive up to 40 degrees Celsius most of the times. Okay, now concentration of substrate. So, as I said, substrate is something which actually activates the enzyme. Without the substrate, the enzyme won't work only. Now, up to a certain level only, they will accept the substrate. So, this is my enzyme. Okay, so this is my enzyme and this is my substrate. Okay, so this is perfect. I have perfect amount of enzyme and perfect amount of substrate and it gets attached. Done. And my enzyme is working at the highest rate, that is the highest velocity, that is Vmax. Now what do? I add more substrate. So this is my enzyme and this is my substrate. This is way bigger than what the enzyme can actually take. Okay, so what will happen? Okay, this will take the substrate, but up to a level only. Up to Vmax only it will take. Like the velocity will up to be Vmax only, up to a certain, after that it is saturated. It won't cross Vmax. Okay, because there is no other enzyme molecule. Enzyme molecule is that much only. There's no other enzyme molecule. So the substrate is there. There are substrate molecule is there, but there is no enzyme to catch it. So there is a saturation here. So always remember, up to a certain limit only, the substrate will attach to the enzyme and work and everything. After that, it is just waste. It will work, but up to that certain Vmax only. After that, it won't work. So concentration of the substrate should be that optimum level only. Again, optimum level, optimum temperature, optimum pH, optimum amount of substrate as well. Okay, next, inhibitor. Okay, so this is something which actually works against the enzyme. Inhibitor itself is actually something like stopping. Inhibition means stopping the reaction. So this one works against the enzymes. So there are particular substances, okay, which actually makes the enzyme stop. They have to stop the reaction of the enzyme. That is where the inhibitors come into play. Okay, so what happens? There are actually three types of inhibitors. The first one is competitive inhibitor. Okay, competitive inhibitor. Okay, so what will the competitive inhibitor do? Okay, I have an enzyme. I have the enzyme. I have the substrate. Okay, let's not take that. Okay, I have an enzyme. I have a substrate. It works. My enzyme works. Now, instead of this substrate, I take this. Okay, this is an inhibitor. Although it looks like the substrate, this is an inhibitor. Okay, so this attaches to the enzyme. Enzyme wrongly, it does not recognize because the structure is the same. It just takes the inhibitor in the place of the substrate. Done. The enzyme is done for. After this, no reaction will take place. The reaction is over. Okay, so that is called competitive inhibitor. Why? Because in place of the substrate, the inhibitor comes and sits. So the reaction is done for. Okay, so inhibitor closely similar to the substrate is called competitive inhibitor. The structure. Okay, and it competes with the substrate. Instead of this, I get this and it attaches. So the inhibitor has won and it will stop the reaction. For example, what? Suppose I have a succinate and there is a succinic dehydrogenase and it forms fumarate. This is a proper reaction. Now, instead of succinic dehydrogenase, sometimes malonate goes and sits because malonate looks a lot like succinic dehydrogenase and the reaction stops. So this is how it works. This is how the competitive inhibitor works. It just goes and attaches and the reaction stops. Okay, so that's the competitive inhibitor. Now, as I said, there are two other inhibitors. There is see, the basic idea is that the reactions should stop. You sit in the place of the substrate, or you sit somewhere else, or you sit with the substrate. That is a different thing. Inhibitor will stop the reaction. Okay, so that was competitive inhibition. Now there is non-competitive inhibition. What will happen here? The inhibitor molecule will just sit somewhere else. Okay, it will sit somewhere else and stop the reaction. That's all. That is what will happen. So this is my substrate and the substrate and enzyme. This is working. Suddenly, this non-competitive inhibitor comes and attaches here. My enzyme is over. My enzyme will not work anymore because this has attached. It won't work. Inhibitor has attached anywhere. This here, some site, it will attach and it will stop working. That is non-competitive inhibition. Now there is uncompetitive inhibition. Okay, what it does is, or anti-competitive inhibition, enzyme inhibitor binds only to the complex formed between enzyme and substrate. So what will happen? It will attach to the complex only. It will attach like this. Not instead of this, not somewhere else. It will attach together. When it has attached together, the enzyme again is done for. Enzyme stops working. Okay, so this is how it works. Now, classification and nomenclature of enzymes. Okay, there are different kinds of nomenclature, different kinds of enzymes. Each and every enzyme has their own work cut out for them. Okay, there are oxidoreductases, which actually catalyzes the oxidoreduction between two substrate. So there is oxidation in one molecule, and reduction in one molecule. Okay. Now there are transferases where one group, any other than hydrogen, one group, okay, some esterase, aldehyde, something will get transferred to another group. Okay, so any one molecule will get transferred to the other molecule. Here we have a G, there's a group, it gets transferred to this S dash position. Now there are hydrolases, okay, this hydrolysis of ester, ether, peptide, glycosidic, halide, PN bonds. Okay, this is hydrolysis reaction, breaking of hydrogen bond. Okay, so lyases, what will lyase do? Catalyze removal of groups by mechanisms other than hydrolysis. Okay, instead of removing a water molecule or anything, it will remove something else. Here it is removal of water molecule or not. Here it removes something else. There are isomerases, catalyze interconversion of optical, geometric or positional isomers like D position or L position, like of glucose, fructose. This is like a position change. Position change in molecules. Okay, and finally ligases, they bring two compounds together. Okay, different parts of enzymes, okay, where they just put different parts of different molecules together. Okay, different parts of DNA, this, that, RNA, different. Ligases have a lot of uses, especially in rDNA technology now. Okay, so brings molecules together. Okay, so finally cofactors. Okay, so these are non-protein constituents bound to the enzyme to make the enzyme active. Okay, so sometimes the enzyme actually needs a special push apart from the substrate. It needs another push by non-protein constituents to actually work properly. They work so beautifully, enzymes work so beautifully. They might be needing a little bit of help. So that kind of help is given by cofactors. So what happens? The apoenzyme is the enzyme in itself, the protein part. Coenzyme is the non-protein part. Okay, so when apoenzyme meets coenzyme, okay, they form a full-fledged enzyme. That is the holoenzyme. This is the proper enzyme. Okay, but when the cofactor is removed, the catalytic activity is lost. Without cofactor, it won't work. Okay, don't get it wrong. Cofactor is not the substrate. Substrate is something else. Cofactor just pushes the enzyme a bit, okay, so that it works nicely. And without it, it won't work. And this is non-proteinaceous in nature. How? See. So there are basically three kinds of cofactors. One is the prosthetic group. Why? Because it is organic and it is tightly bound to the apoenzyme. So there is an organic group. Okay, this is non-proteinaceous, but it is organic in nature, and it will bound tightly, very tightly to the apoenzyme. It is a part of active site of the peroxidase, catalase, etc. See here, this is the prosthetic group, and it is bound tightly to the enzyme, and it forms the holoenzyme. Okay, now there are coenzymes. What happens? This is also organic, but this is transient binding, not that tightly. Okay, that the prosthetic group is tightly bound. This is just like that. It is just bound, and it just pushes the apoenzyme a bit. Okay, that is the coenzyme. And finally, there are metal ions. They form, this is inorganic in nature. Okay, they form coordination bonds with the side chains at active site and one or more coordination bonds with the substrate. They just form bonds. Okay, they form bonds with the apoenzyme. Okay, so that is how it works. Okay. Okay then. So if you have any doubts, put it in the chat box. Let's just solve some questions and we can leave it today. Okay. An enzyme of competitive inhibition of an enzyme, of an example of competitive inhibition of an enzyme is the inhibition of succinate dehydrogenase by malonic acid, cytochrome oxidase, hexokinase, and carbohydrate anhydrase. Okay. So competitive enzyme, competitive inhibitor, what happens? So there is an enzyme, okay, and instead of the substrate, okay, another molecule, okay, the inhibitor molecule comes and sits, such as we gave an example, succinate dehydrogenase. So succinate dehydrogenase, instead of that, malonic acid comes. So that is how it works. Next, enzyme increase the rate of reaction by, so what I said, you need a little bit of push, you need that activation energy is required to work, right? So we need the activation energy. So when we put the activation, when we put the enzyme, we don't need that much energy, we need just a little push because we have the enzyme. So we need lower amount of activation energy because we have the extra push from enzyme, no? So we need lower amount of activation energy. So lowering of activation energy. Okay. Okay. So with reference to enzymes, which of the following is true? So we know we have apoenzyme, which is the protein part, and there is the coenzyme, which is the non-protein part, which forms the holoenzyme, that is the full enzyme. So see, holoenzyme is apoenzyme plus coenzyme. So it is option B. Okay. Next, the fastest enzyme known as, we know this, it is carbonic anhydrase, where it formed 600,000 molecules of H2CO3, that is carbonic acid, okay, in an hour. Okay. So that's it for today. In the next class, we'll talk a little bit about cells and everything. Okay. Don't forget to like, share, and subscribe to our channel. You can even try our new channel as well, which is solely dedicated for 11th and 12th standard students. Okay. So I'll see you guys in the next class. Bye-bye.